Key dopamine pathways in the brain. The 5 dopamine pathways in the brain are shown in Figure 4 to 11. They include the mesolimbic dopamine pathway, the mesocortical dopamine pathway, the nigrostriatal dopamine pathway, the tuberoinfundibular dopamine pathway, and a fifth pathway that innervates the thalamus. The dopamine hypothesis of schizophrenia. The mesolimbic dopamine pathway and positive symptoms of schizophrenia. The mesolimbic dopamine pathway projects from dopaminergic cell bodies in the ventral tegmental area of the brainstem to axon terminals in one of the limbic areas of the brain, namely the nucleus accumbens in the ventral striatum, Figure 4 to 11. This pathway is thought to have an important role in several emotional behaviors, including the positive symptoms of psychosis, such as delusions and hallucinations, Figure 4 to 12. The mesolimbic dopamine pathway also is important for motivation, pleasure, and reward. For more than 40 years, it has been observed that diseases or drugs that increase dopamine will enhance or produce positive psychotic symptoms, whereas drugs that decrease dopamine will decrease or stop positive symptoms. For example, stimulant drugs such as amphetamine and cocaine release dopamine, and if given repetitively can cause a paranoid psychosis virtually indistinguishable from the positive symptoms of schizophrenia. Stimulant drugs are discussed in detail in subsequent chapters on treatment of attention deficit hyperactivity disorder and on drug abuse. All known antipsychotic drugs capable of treating positive psychotic symptoms are blockers of the dopamine D2 receptor. Antipsychotic drugs are discussed in Chapter 5. These observations have been formulated into a theory of psychosis sometimes referred to as the dopamine hypothesis of schizophrenia. Perhaps a more precise modern designation is the mesolimbic dopamine hypothesis of positive symptoms of schizophrenia, since it is believed that it is hyperactivity specifically in this particular dopamine pathway that mediates the positive symptoms of psychosis, Figure 4 to 13. Hyperactivity of the mesolimbic dopamine pathway hypothetically accounts for positive psychotic symptoms, whether those symptoms are part of the illness of schizophrenia or of drug-induced psychosis, or whether they are positive psychotic symptoms accompanying mania, depression, or dementia. Hyperactivity of mesolimbic dopamine neurons may also play a role in aggressive and hostile symptoms in schizophrenia and related illnesses, especially if serotonergic control of dopamine is aberrant in patients who lack impulse control. Although it is not known what causes this mesolimbic dopamine hyperactivity, current theories now state that it is the downstream consequence of dysfunction in prefrontal cortex and hippocampal glutamate activity, as will be discussed below. The mesocortical dopamine pathway and cognitive, negative, and affective symptoms of schizophrenia. Another pathway also arising from cell bodies in the ventral tegmental area, but projecting to areas of the prefrontal cortex, is known as the mesocortical dopamine pathway, Figures 4 to 14 and 4 to 15. Branches of this pathway into the dorsolateral prefrontal cortex are hypothesized to regulate cognition and executive functions, Figure 4 to 14. Whereas branches of this pathway into the ventromedial parts of the prefrontal cortex are hypothesized to regulate emotions and affect, Figure 4 to 15. The exact role of the mesocortical dopamine pathway in mediating symptoms of schizophrenia is still a matter of debate, but many researchers believe that cognitive and some negative symptoms of schizophrenia may be due to a deficit of dopamine activity in mesocortical projections to dorsolateral prefrontal cortex, Figure 4 to 14, whereas affective and other negative symptoms of schizophrenia may be due to a deficit of dopamine activity in mesocortical projections to ventromedial prefrontal cortex, Figure 4 to 15. The behavioral deficit state suggested by negative symptoms certainly implies underactivity or lack of proper functioning of mesocortical dopamine projections that may be the consequence of neurodevelopmental abnormalities in the NMDA, N-methyl D-aspartate, glutamate system. Described in the next section. Whatever the cause, a corollary to the original DA hypothesis of schizophrenia now incorporates theories for the cognitive, negative, and affective symptoms, and might be more precisely designated as the mesocortical dopamine hypothesis of cognitive, negative, and affective symptoms of schizophrenia, since it is believed that it is underactivity specifically in mesocortical projections to prefrontal cortex that mediate the cognitive, negative, and affective symptoms of schizophrenia, figure 4-16. Theoretically, increasing dopamine in the mesocortical dopamine pathway might improve negative, cognitive, and affective symptoms of schizophrenia. However, since there is hypothetically an excess of dopamine elsewhere in the brain, within the mesolimbic dopamine pathway, any further increase of dopamine in that pathway would actually worsen positive symptoms. Thus, this state of affairs for dopamine activity in the brain of patients with schizophrenia poses a therapeutic dilemma. How do you increase dopamine in the mesocortical pathway while simultaneously decreasing dopamine activity in the mesolimbic dopamine pathway? The extent to which atypical antipsychotics have provided a solution to this therapeutic dilemma will be discussed in Chapter 5. Mesolimbic dopamine pathway, reward and negative symptoms. When a patient with schizophrenia loses motivation and interest, and has anhedonia and lack of pleasure, such symptoms could also implicate a deficient functioning of the mesolimbic dopamine pathway. Not just deficient functioning in the mesocortical dopamine pathway. This idea is supported by observations that treating patients with antipsychotics, particularly the conventional antipsychotics, can produce a worsening of negative symptoms and a state of neurolepsis that looks very much like negative symptoms of schizophrenia. Since the prefrontal cortex does not have a high density of D2 receptors, this implicates possible deficient functioning within the mesolimbic dopamine system, causing inadequate reward mechanisms exhibited as behaviors such as anhedonia and drug abuse, as well as negative symptoms exhibited as lack of rewarding social interactions and lack of general motivation and interest. Perhaps the much higher incidence of substance abuse in schizophrenia than in healthy adults, especially of nicotine but also of stimulants and other substances of abuse, could be partially explained as an attempt to boost the function of defective mesolimbic dopaminergic pleasure centers, perhaps at the cost of activating positive symptoms. Nigrostriatal dopamine pathway. Another key dopamine pathway in the brain is the nigrostriatal dopamine pathway, which projects from dopaminergic cell bodies in the brainstem substantia nigra via axons terminating in the basal ganglia or striatum, figure 4-17. The nigrostriatal dopamine pathway is a part of the extrapyramidal nervous system and controls motor movements. Deficiencies in dopamine in this pathway cause movement disorders including Parkinson's disease, characterized by rigidity, akinesia, bradykinesia, i.e., lack of movement or slowing of movement, and tremor. Dopamine deficiency in the basal ganglia also can produce akathisia, a type of restlessness, and dystonia, twisting movements especially of the face and neck. These movement disorders can be replicated by drugs that block D2 receptors in this pathway, and this will be discussed briefly in Chapter 5. Hyperactivity of dopamine in the nigrostriatal pathway is thought to underlie various hyperkinetic movement disorders such as chorea, dyskinesias, and tics. Chronic blockade of D2 receptors in this pathway may result in a hyperkinetic movement disorder known as neuroleptic-induced tardive dyskinesia. This will also be discussed briefly in Chapter 5. In schizophrenia, the nigrostriatal pathway in untreated patients may be relatively preserved, figure 4 to 17. Tuberoinfundibular dopamine pathway. The dopamine neurons that project from hypothalamus to anterior pituitary are part of the tuberoinfundibular dopamine pathway, figure 4 to 18. Normally, these neurons are active and inhibit prolactin release. In the postpartum state, however, the activity of these dopamine neurons is decreased. Prolactin levels can therefore rise during breastfeeding so that lactation will occur. If the functioning of tuberoinfundibular dopamine neurons is disrupted by lesions or drugs, prolactin levels can also rise. Elevated prolactin levels are associated with galactorrhea, breast secretions, amenorrhea, loss of ovulation and menstrual periods, and possibly other problems such as sexual dysfunction. Such problems can occur after treatment with many antipsychotic drugs that block D2 receptors and will be discussed further in Chapter 5. In untreated schizophrenia, the function of the tuberoinfundibular pathway may be relatively preserved, figure 4 to 18. Thalamic dopamine pathway. Recently, a dopamine pathway that innervates the thalamus in primates has been described. It arises from multiple sites, including the periaqueductal gray matter, the ventral mesencephalon, various hypothalamic nuclei, and the lateral parabrachial nucleus, figure 4 to 11. Its function is still under investigation, but it may be involved in sleep and arousal mechanisms by gating information passing through the thalamus to the cortex and other brain areas. There is no evidence at this point for abnormal functioning of this dopamine pathway in schizophrenia.